Good evening to everyone. Well, at least here it's evening. I don't know where you're dialing in from, what time it is, but good evening. Thank you for inviting me to talk in this seminar. I'm actually very excited to join and be able to share with all of you our recent work in engineering synthetic communication in mammalian cells, as you see the title of this slide. My name, very nicely and correctly said, is Anna Maria Makri Pistikou. I know it's a mouthful. And I am indeed a doctoral candidate in Eindhoven University of Technology, more specifically in the Institute of Complex Molecular Systems, and the university is in the Netherlands. To make it a little bit more personal, and since we have plenty of time, I want to start this seminar by telling you a bit about myself before we dive into the hardcore science. I originally come from Athens, Greece, therefore my name. In 2009, I moved to Sparta to get a nursing and bachelor's degree from the University of Peloponnese. Pretty quickly, I realized that taking care of people is not really my calling. And believe it or not, this is quite a prerequisite to be a good nurse. So I moved to the Netherlands to do another study. So in 2016, I graduated from Maastricht University with a bachelor's degree in cell and molecular biology. And in the same university, I did a research master's in cognitive and clinical psychology with specialization to fundamental neuroscience. There I studied the role of microRNAs in neurodegenerative disease, so a little bit of neuroepigenetics. In 2018, though, I moved to Eindhoven, which is the Dutch city of light, as you see nicely from this picture, where I started my PhD trajectory in mammalian synthetic biology. So a little bit of change of gear. A big part of the work that that I did during my PhD, I actually would like to share with you today. And I just want to mention up front that this work was recently published in Nature Communications. One more important aspect that I would like to mention here is that this is a collaborative effort, as you see from the quite big author list on the paper. But I really want to take a little bit of time to acknowledge the blue shirt team. As you see here, the color coordination was done completely by accident, but I always find it a little bit funny. And that is Dr. Glenn Kremers, engineer Brian Natalia, and of course, Professor Tom de Graef, which is my direct supervisor. Now on to the science. The broader field that the research I'm going to talk to you about is falling under is the cell-based therapeutics field. What revolutionized the field in the last decades is the novel functions. This is one of the key aims of synthetic biology, as I'm sure you know. And for those that you don't know what cell-based therapeutics really is, well, the concept, it's fairly simple. In a nutshell, you isolate cells from a patient or a donor, engineer the cells to make them responsive to certain disease, expand them, and put them back into the patient to fight that specific disease. In particular now, the introduction of synthetic receptors has enabled the engineering of designer cells that can sense and correct disease state. But so what are synthetic receptors? Our cells have the innate ability to sense their environment and respond to it, and they do that by engaging cell surface receptors. For example, here you see T cells or TCR that upon binding to its cognate ligand, something that is usually presented to the major histocompatibility complex, MHC, is able to activate the T cells and trigger the immunological response. Now, scientists realized early that the functionality of those self-surface receptors can be harnessed to sense and correct for disease state. And in that way, they built the first synthetic receptor, namely the chimeric antigen receptor, or CAR, that you see here. And what basically was done very smartly is that the extracellular domain of the T cell receptor, the native T cell receptor, was exchanged for a single chain variable fragment, as you see here in red, that can recognize a disease antigen independent of the MHC, the major histocompatibility complex. And here you see a very simplified representation of the concept. CARs are engineered to redirect the specificity of immune cells, typically T cells, but others have been engineered towards specific antigens found on the target cells, such as cancer cells. CAR T cell therapy has demonstrated remarkable success in clinical trials, leading to durable remissions and even cures in some patients with certain types of leukemia and lymphoma. This is all hematological blood tumors, hematological disease. In detail, there are in total 6 FDA-approved CAR T treatments that you see in the little table that I made here. So thinking back on the schematic I showed you before, T cells can be isolated from a patient that suffers, for example, I don't know, from a large B-cell lymphoma, one type of hematological cancer. These cells are engineered to express a CAR, in this case, that is sensitive to an antigen present on the surface of the tumor cell. They are and then they are reintroduced into the patient's bloodstream. When CAR T cells encounter cells that express target antigen, the extracellular targeting domain, as you see here, binds to the antigen that the CAR is directed towards, triggers the intracellular signaling cascades through the intracellular signaling domain, and this activation leads to the destruction of the target cell. That happens through various mechanisms, including, for example, the release of cytotoxic molecules and cytokines, induction of apoptosis, and recruitment of other immune cells. Of course, CARs are not the only synthetic receptors with ambitions to treat disease. One other great example, my personal favorite, is the synthetic Notch receptor that was developed by the group of Lim in 2016. And you see like a very, very, very schematic representation here. This receptor now is engineered not only to sense a custom input, as you see with the red color extracellularly, but also to result in a custom defined output. Simple principles of engineering again. In the C-notch, not only the extracellular domain of the natural notch was exchanged, but also the intracellular domain too, with a synthetic transcription factor, resulting in programmable responses. Additionally, a number of other synthetic receptors have been engineered, amongst which the GEMS receptor that is able to respond to soluble stimuli. I do want to talk a little bit more about the GEMS receptor here, since this is the platform we use to engineer communication in mammalian cells. The GEMS basically retains its signal tranquilities by incorporating the erythropoietin receptor transmembrane domain, the EPO-R that you see here, and that has been mutated to be inert to erythropoietin, so it doesn't respond to the native ligand. GEMS can then equipped with an extracellular domain, and that can recognize a bunch of different antigens, soluble antigens, that they showed in the original paper to cover inputs ranging from really small to really big proteins, well, bigger. The EPO-R transmembrane domain was also fused to the intracellular signal transduction domains of some known receptors. And once the receptor is activated through dimerization, these intracellular domains induce downstream signaling, different intracellular pathways. Minimal promoters are then that are selectively responsive to these pathways are used, and they can rewire the signaling to a transgene expression. So you can express a transgene of choice. It sounds great, doesn't it? Well, it isn't that great. Actually, there are a number of challenges that are associated with cell-based therapeutics. One major challenge is that each engineered cell can only harbor a small number of synthetic functions because of the high metabolic burden due to the expression of multiple heterologous processes. If you load up a cell with extra genes that you want them to express, yeah, then they're not going to manage at the end. But yeah, okay, why do we need to introduce more advanced functions? Something I did not tell you is that on all these therapies, there is a high risk of development of adverse effects. One example is on target off-tumor toxicity. What do I mean by that? That can happen when engineered cells recognize and attack target cells expressing the target antigen at low levels on healthy tissues. One approach to solve this would be to introduce combinatorial recognition of antigens on the target cell. As you see, for example, like very simple schematic here. So 2 or more antigens must be recognized on that diseased cell in order to trigger the response. But as I said, this is quite challenging since it is associated with really high metabolic burden. So in order to be able to realistically achieve advanced functions, you need distributed functions. For example, as you see in this schematic here, you can have one cell population that is wired to sense input 1, a second cell population that is wired to sense input 2, and a third cell population that is able to integrate these responses and execute the desired function. And to do that, you need to introduce a synthetic communication system amongst your engineered cells that is ideally bi-orthogonal to the natural environment. You don't want it, of course, to interact with the natural processes. To realize now intercellular communication and enable programmable control of neighboring cells and neighboring cell responses, ideally, cells should be able to sense bi-orthogonal, as I said, but also soluble stimuli that is scalable, while at the same time being able to produce user-defined responses, as you see here. Additionally, such a system, it would be great if we could enable distributed computing that allows the performance of logic operations. And that is exactly what we wanted to do. And for that purpose, we exploit the GEMS platform that I a little bit briefly introduced in a previous slide. It was developed by Fusenegger and his team in 2018. And that uses, I will repeat because it's quite important, the inert version of the erythropoietin receptor, EPO receptor, to detect soluble stimuli. And when it dimerizes, it's also quite important, it can result in a custom-defined output. Here you see the activation of the GEMS, the original GEMS receptor, via 2 distinct intracellular pathways, as was published in this paper, namely the JAK-STAT pathway, you see here, and the MAPK for different concentrations of RR120. This is like the soluble ligand recognized by this specific GEMS receptor. So what did we do? We functionalized the receptor with previously designed coil peptide pairs that have the ability to orthogonally bind to a select partner. You see them here, the coils, they turn. So as you see in this inset, A only binds to A prime, B only, I will call it alpha, only binds to alpha prime, beta only to beta prime, and C or gamma only binds to gamma prime, and so on. And the nice thing about these coil pairs is that they have the potential to be scalable, with people showing with a nice paper on Nature in 2019, a bunch of pairs. They showed basically programmable design of a large set of these coil pairs. So first, of course, proof of principle. Can you design CC-GEMS receptors, that's what we call them, to be activated by a designer ligand that consists of 2 coil pairs spanned by a linker? Of course, you want 2 pairs because you need to dimerize a receptor. Next, can you harness the built-in programmability of the CC-GEMS to demonstrate Boolean logic operations and ultimately achieve this intercellular communication between, for example, very simply shown here, a sender cell that sends the ligand and a receiver cell that receives the ligand and gives a response. Initially, we want to showcase activation by receptor heterodimerization of a cognate coil pair. So does it even work to activate these receptors with these coils? We didn't know. And here in the schematic, you see a very typical setup for such an experiment, where you have, where we chose 293T mammalian cells, were also used in the original GEMS paper. And we transiently transfect them with 4 plasmids. You see them here. 2 plasmids that encode the receptor heterodimers, in this case that you see here, cognate receptor heterodimers, alpha and alpha prime GEMs, and in this case utilizing the JAK-STAT pathway. Then 1 plasmid encoding STAT3 and 1 for the reporter gene, STAT3 induced secretin alkaline phosphatase, or I will from now on call it CIAP. This is our reporter gene. Following receptor dimerization and activation, therefore, STAT3 is phosphorylated and becomes a transcription factor for the CIAP reporter gene. To put it very simply, CIAP expression means receptor activation. CIAP activity is then quantified after hydrolysis of P9. I know it's a lot of difficult names, but basically that can be measured on a spectrophotometer. It gives color with a commercially available kit. And here you see the kinetic trace in this little graph of substrate cleavage from the enzyme, the CIAP enzyme, in micromolars over 30 minutes. And you can very clearly see that the receptor pair that can heterodimerize due to the functionalization with cognate alpha and alpha prime cold-coil pairs results in receptor activation, so the blue line. The non-cognate, in this case, the alpha beta GEMS receptor that is not supposed to dimerize, indeed did not result in substrate cleavage, therefore reporter expression. We also investigated whether receptor activation is dependent on the linker length between the CC domain, the cold-coil domain, and the transmembrane scaffold on the receptor. You see it here. I call this alpha linker. Yes, it's all Greek, of course. I am Greek, so makes sense. We engineered now alternative CC GEMS receptors with varying linker lengths, alpha of 0, as you see here, the color coding corresponding to the different linkers. I hope it's clear. 4 amino acids, 8 amino acids, and 27 amino acids in length. And what you see from this graph, and I hope you can appreciate, is first a verification of the orthogonality of all the alternative alpha-alpha prime pairs. As you see, cognate pairs results in reporter expression, while non-cognate pairs don't. So alpha-alpha, which is not supposed to bind, or alpha prime, or alpha beta does not give a response, while all the alpha-alpha prime pairs do. And what you can also see is that, at least for these linkers, receptor activation seems to be quite robust, since there is no difference in receptor activation level. So, okay, this is now step 1, proof of concept on how cold-coil interactions can be used as a robust and programmable tool to activate synthetic receptors. Still, I remember the day that we got these results. I was very excited, many years ago. Next, we want to see if an engineered ligand, what you see a schematic here, composed of the 2 cold coils, would also trigger receptor activation. So we need to engineer and express a dipeptide alpha prime cold coil ligand. To start, we express the alpha prime cold coil, a monomer, only 1 peptide, in a bacterial expression system after sumo tagging it. We engineered the cold coil in such a way as to contain a single cysteine. Purification steps. Since these things bind in parallel, to produce a ditopic ligand, we link the N-termini of these monomers of the alpha prime CCs using maleimide chemistry. You see the results, like the end results here. So you see that we managed to engineer the ditopic alpha prime cold coil ligand. Here you see that when titrating the synthetic ligand, the dipeptide, alpha prime, on the cells expressing the cognate alpha GEMS receptor, we see reporter expression that is indicative of a dose response curve. Additionally, you see here a quite broad plateau that ranges between 0.015 to 0.5 micromolars of titrated ligand. And as expected, the addition of monomeric alpha coil in the culture medium did not result in activation, which is similar to the condition where no ligand was added. So as you see here, 0. Again, here we checked for the receptor activation levels caused by the addition of the alpha prime dipeptide ligand on the medium of 293 T cells that express cognate alpha GEMS for various linker lengths alpha between the coil and the transmembrane part of the receptor. And again, as you see, and you can appreciate, I think here, all cognate alpha GEMS receptors were activated by alpha prime dipeptide ligands, while the non-cognate beta GEMS were not. However, facilitating synthetic communication using CC-GEMS is our ultimate purpose here. So comes without saying, I think, that the ligand-mediating receptor activation should be at the end expressible by mammalian sender cells. And something that is N-terminal linked, at least to my knowledge, cannot be expressed. So we devised a strategy that you see here in the insert, an alternative to our previous strategy, where we connected the C-terminus of one coil to the ends of another. And in that way, the design ligand can be expressed in mammalian cells. There is, of course, an expected minimum length that such a linker needs to be able to bridge in order to facilitate the activation. So we tested 3 different types of linkers made out of combinations of more rigid alpha helical blocks and more flexible SGS repeats, SGS domains. And for the sake of clarity, I will refer to these linkers from now on as L1, L2, L3. I don't know if you ever tried to express something in mammalian cells, but trust me, it is not the easiest. There's really low yields. So first, expressed always in E. coli. And here you see all the STS pages showing the expressed ligands for the different ligands having the different linkers for L1, L2, and L3. Next step, can these ligands activate the receptor? The answer is yes. These ligands can activate the cognate alpha GEMS. We opted from now on for a linker length between the EPOR and the coil of alpha-2, like 8 amino acids. And we see here that the longer linker, L3, the ligand results in a relatively lower activation. But what was interesting here is that the presence of or absence of the SUMO tag does not seem to hinder activation, which is very convenient for our later attempt to establish intercellular communication because we don't have to deal with it. To verify the coil-coil ligand receptor interaction binding, we labeled the SUMO tag alpha prime with fluorescent dye, Cy3 in this case, and incubated with 293 T cells that you see here, the little cartoon that express the cognate alpha GEMS with this linker that I talked about of 8 amino acids. We did that for 24 hours, and you see here the confocal fluorescence microscopy. And I think you can appreciate that you have ligand binding, and that indirectly confirms the presence of the alpha GEMS on the cell membrane and validates their ligand receptor interaction. And as expected, when we label the ligand and incubate it with cells expressing non-cognate receptors, as you see here, beta GEMS and gamma GEMS, you see no binding. But so what do we know so far? CC functionalized, call functionalized GEMs, CC GEMs receptors, wired to signal through the JAK-STAT pathway, that's what we are using so far, can be activated through the binding of receptor dimers to a cognate dipeptide called ligand that can be expressed. That's what we know so far. So here I have a little bonus. We showed also that CC GEMs, similar to original GEMs receptor, can also signal through an alternative PLCG pathway. I will give you a little bit more detail here, although I might bore you. We fuse the alpha call functionalized EPOR transmembrane scaffold on the GEMs receptor to the intracellular signaling domain derived from the VEGFR2. When the receptor basically dimerizes through the PLCG pathway, basically, and that can result in the expression of the reporter gene, CEP, in this case, using the NN-FAT minimal promoter, and this promoter is responsive to this path. Another little bonus here. So far, I'm showing you how CC GEMs can be used for the expression of our reporter gene, CEP. Very nice, very easy to measure. But here you see that with not a lot of surprise, of course, how CC GEMs activations from a cognate dipeptide ligand can also result in an expression of a different thing, a different protein, in this case, interleukin-10, IL-10. Okay, that was a little detour, but going back to the original plan and all of using the JAK-STAT pathway and our reporter, our favorite reporter. What is great about this platform? Well, as I mentioned at the very beginning of my talk, is that it has the potential to be scalable. And theoretically, that is, of course, the case, since you have all of these nicely shown to be orthogonal call pairs. But although theory is great, I tried to convince scientists or reviewers that claim holds based on theoretical predictions. Well, good luck with that. So, of course, we want to show evidence of this anticipated scalability. And again, it's proof of concept. And to do this, we expressed two alternative sumotagged ditopic call ligands with linker L2. It opted for one linker. You see here the beta prime, and gamma prime, again in E. coli, again in bacteria. We labeled these ligands with fluorescent dyes and incubated with 293 T cells that express their cognate receptor. And you see the results of the confocal here. You see ligand binding to the receptor. But what happens to the reporter gene expression? We saw binding. As expected, only cognate ligand receptor pairs result in expression of the reporter. So you see here that only the alpha prime ditopic ligand can activate the alpha GEMs, while the non-cognate beta prime, and gamma prime result in no activation. And that holds true for the beta GEMs receptor that is only activated by beta prime ligand, and gamma GEMs. You also see here the results that is only activated by gamma prime. Little fun fact, interesting, again, I think, incubating alpha GEMs receptor expressing cells with an equimolar mixture of alpha prime, beta prime, and gamma prime ligands, this bar on the right, exhibits similar activation levels, well, basically the same, when you incubate with alpha prime solely alone. And that, I think, shows very nicely that non-cognate cold dipeptides do not inhibit cognate dipeptide receptor binding. I don't know how they would, since we show that they do not bind. The inheritor of those cognate coils makes CC GEMs an ideal platform for the implementation of Boolean logic, which is one of my favorite features. For example, engaging an array of CC GEMs and dipeptide CC ligands has the potential to enable receptor activation or gate operations, or as you see here, combinatorial ligand sensing, so NGLIT. So let's begin with the construction of the OR gate, which was the most tricky because this requires, as you see in this little schematic, the expression of 3 unique CC GEMS receptors. So you see the example here. You need basically a mammalian cell expressing on its surface alpha GEMS, beta GEMS, and gamma GEMS. And you need also to construct 2 unique dipeptide cold coil ligands. And in this design that I have here, an alpha prime, beta prime, and an alpha prime, gamma prime dipeptide ligand. We express those in E. coli, again, purify them. And what you see here is that sumo-tagged beta prime, alpha prime ligand can activate the beta alpha, I guess, GEMS receptor heterodimer. While similarly, the alpha prime gamma prime ligand can activate the alpha gamma receptor heterodimer. And finally, the presence of both ligands results in reporter expression, so activation. Now for the AND gate, bear with me. We need to express 2 unique ditopic cold coil ligands, as you see here, the alpha prime gamma and the alpha prime gamma prime dipeptide ligand. And this design, and what you see in this inset, the gamma prime gamma pairing is expected to facilitate interligand dimerization, resulting in a cold coil complex with, you see here, the green, yellow, mustard yellow, green, dark green again. And this complex has 2 accessible alpha prime domains. Those alpha prime domains, the dark green, are available to bind to alpha GEMS receptors, the more light green. We express these ligands again in E. coli, in bacteria, and as expected, you see in the absence of input, there is no reporter expression, as well as in the presence of either alpha prime gamma or alpha prime gamma prime ligand alone. However, when you incubate 293 T cells that express the alpha GEMS with both alpha prime gamma and alpha prime gamma prime, you see a sharp increase here, the blue bar, in reporter expression that shows receptor activation. On to engineering synthetic communication. What is the simplest form of communication between 2 cells you can think of? I guess you will agree with me that it involves a sender cell, as I show like a very basically here, that can secrete a signaling molecule, in our case, the dipeptide, that can activate a receiver cell. But so far, we have expressed all the ligands in bacteria. So you guessed the natural next step. The answer we needed to give was, can we express ligands in mammalian cells? Yes, we can. You see it here. We know that the presence of sumo-tagged doesn't seem to be a problem. So the only bottleneck is, can mammalian cells express it? And if they can express it, can this ligand activate cognate receptors? And the answer to the first question, you see already, I already talked about it. Yes, you can express it. And yes, it can activate cognate alpha GEMS receptors. Beta, as you see, like a little bit here in a bit moderately decreased levels compared to when you compare to the ligand produced in bacteria. To engineer sender cells, we added 1 more layer of complexity because why not? We wanted to achieve transcriptional control over the ditopic ligand expression. So we placed the sumo-tagged alpha prime gene under the control of a doxycycline-induced CMV promoter. And what you see here, apologies if it's too small, is that an emerald green fluorescent protein is expressed as a bicystron under the same promoter through the utilization of an IRES self-cleaving peptide. Probably you know it. We antiviral induced 293S, DNTI, theta R cells, a lot of jargon, I know, with the transgene. But basically what you need to know to understand how we manage to engineer the communication is that these cells constitutively express theta R that combine to the promoter and repress expression of the transgene. So simply said, these cells will not express the transgene in the absence of doxycycline. So no doxycycline, no ligand secreted. And as it follows, no GFP expression, it's a bicystron. But you see here that when you add doxycycline, the bottom panel on the flow cytometry results, in the culture medium of these cells, the ligand is expressed. And you see that from the expression of GFP. Then we did, of course, a co-culture. So in the co-culture of senders, T cells that express the alpha gems, we see a sharp increase, blue bar, in reporter gene expression with the addition of doxycycline. So here's the inducer. Well, this is not the case in the absence of the inducer. So when there's senders but no doxycycline, this is also not the case when we have control cells that do not express the ligands. So engineering synthetic communication through CC gems seems to be possible. I hope I have convinced you with this data, and can even be transcriptionally controlled with an addition of the inducer, in this case, the doxycycline. I think you kind of guessed what we want to do next, which is to establish a minimal free cell population system. You see the schematic here faded. That together can execute the NGATE logic. For this, we needed two types of sender cells populations. One population that expresses the alpha prime gamma ligand, let's call them senders 1 quite unimaginatively. And a second cell population expressing alpha prime gamma prime ligand, we call them senders 2. And remember, gamma prime gamma interligand dimerization, I saw it here in the insert, results in the, bear with me, alpha prime gamma alpha prime complex. So green, yellow, green. That has the two accessible alpha prime domains that are available for binding the cognate alpha gems receptor. And of course, what you need to make this communication system possible is the receiver cell that expresses the alpha gems. Senders 1, check, can express ligand. Same goes for senders 2. And when you co-culture the three populations together, you see reporter gene expression, blue bar again. Blue is my favorite color. While this is not the case for a co-culture with one of the ligand expressing populations and the receivers. So a little conclusion. By leveraging the ability of these cold coils to exclusively bind to a select partner, I hope I have convinced you that we could show orthogonal receptor activation, as well as Boolean logic computations. And finally, by designing a cold coil dipeptide that can be expressed in mammalian cells under the control of an on-off switch, we achieved intercellular communication. I think you can appreciate that the work that I presented to you today involves a considerable amount of engineering, which would not have been possible without the help of certain people. Well, okay, maybe it would have been possible, but it would definitely take me more than a decade to conclude. So I would like to take 1 more minute from your time to acknowledge Glenn, Brian, for all their help in designing experiments and in the lab, the power horses, and of course Tom, without whom this research wouldn't have been possible, as he's the one with all the crazy and creative ideas. But also the rest of the people, the names you see written down here, Tom, Bas, Bruno, Anne, Martin, Oscar, Carlene, second Martin, and Roman. And of course the funding bodies, since I was not the one paying for all of this, and trust me, it's quite expensive. And I would like to also thank you for your attention, and I would be happy to answer questions, I think.